Welcome back, everybody, to NECAFS 2021. Appreciate you all joining us again for the second day, for some of you, and the third day for others. Great to have you back. And I wanted to start out today by, first of all, just appreciating the amount of thought and effort and work that went into having the conversations you did yesterday. Elizabeth and I got emails last night from Google saying, slow down, we didn't expect Google Docs to be used so much in one day, but it was great that all came together, and appreciate you all being flexible with using that approach. I want to start out this morning with some thank yous, and to begin with, we're grateful to the USDA NEFA Food Safety Outreach Program for providing funding for NECAFS that's supporting the current meeting. I also want to say thank you to the FDA for our original funding that helped start up NECAFS. And when we first got started. And then the other thank yous that we have today are to start with the project team. So forgot to mention this yesterday, but NECAFS has a PI team that includes not only Elizabeth and I, but also Betsy Ben from Cornell and the PSA, Amanda Kinchler from University of Massachusetts, and Luke Laborde from Penn State. This was the planning committee for the regulator one-day meeting that was actually day one of the NECAFS meeting this year, where the regulatory group was able to get together and talk about some of the observations they've had that actually fed into the discussions yesterday. So thank you to all these folks who represent the state CAP grant group. They also had some breakout facilitators during that meeting. And again, really appreciate everybody who has volunteered this year or who was voluntold to serve as a facilitator for one of the breakout groups. These are really important roles. And I realize it's a stretch for some folks, and a stretch just in general to try to bring a group together in an intentional discussion. So thanks for doing that. Same with the PC meeting. So NECAFS annual meeting kind of has this parallel track of preventive controls and produce safety, breakout groups and meetings. And again, appreciate the folks who have stepped up this year to keep the preventive controls meeting moving along and facilitated. We also have work group leads for the preventive controls group. So they've sort of been able to establish more of a continued structure during the year. And so these are the folks that head up those sustained work groups throughout the year. And yesterday on the produce safety side, we also had some breakout group facilitators. And thank you again to these folks who were able to help keep the conversations moving and focused, and also capture the discussions. With that, again, just a general thank you to everybody, and a special thank you to Elizabeth Newbold for, as always, keeping us organized and getting this meeting pulled together. And with that, I'll turn it over to Elizabeth.
Thank you, Chris. I now have the pleasure of introducing our speakers for this morning, and I want to point out that we will have a Q&A roundtable after the presentations are over. So please utilize the chat function on the video cast in order to put your questions into that box. And after the presentations are over, we will have a Q&A. So please take that opportunity and put any questions you have into that chat box. And so to begin, I'm pleased to introduce Adriana Jimenez with the FDA Produce Safety Network. Adriana recently joined the CIFSAN Produce Safety Network in December, and she is based in the Division of Produce Safety. She took over the work for Socrates as the PSN representative in the Northeast region. Adriana is located in Northern Virginia on the border of West Virginia. And so we are excited to welcome Adriana, and now I'll turn it over to her presentation.
Good morning, everyone, and thank you for providing us with some time today to share an update on behalf of the Fresh Produce Branch, the CIFSAN Produce Safety Network. I wanted to start by giving a quick introduction of myself. My name is Adriana Jimenez Lopez, and as we are now probably aware, I just joined the CIFSAN Produce Safety Network. I'm based within the Division of Produce Safety, and I will be working with Fresh Produce Safety in the Northeast region. I took over Socrates Trujillo. He used to be your CIFSAN contact in the region, and I'm physically located in the Northern Virginia along the border of West Virginia. So previous to joining the Product Safety Network, I was an FDA ORA domestic investigator within the Baltimore, Maryland district for about 6 years. And during that time, I worked mainly on the field, conducting different food manufacturing establishment inspections and investigations. Where during that time, I specialized and conducted different use hazard inspections and animal food preventive control inspections. Prior to FDA, I worked with the USDA, the Natural Resource Conservation Service, as a soil conservationist in the state of West Virginia. During my time with NRCS, I worked directly with farmers in helping them developing effective soil and water conservation plans, promoting good agricultural practices, and addressing resources concerns related to soil, water quality, and plants. I'm originally from Puerto Rico, where I obtained my bachelor and master's degree, both from the University of Puerto Rico in agricultural sciences and crop protection. So that's some of my background. I hope we can soon connect to talk about different programs or projects that you're working on and see where we can find any opportunity to further collaborate more in as regards of fresh produce safety. So moving on with the presentation. All right. So the Product Safety Network team, we all work remotely. We have members located all around the country. That enables us to develop and utilize local knowledge about crops, environment, and unique challenges in each area. The PSN is based in the Division of Produce Safety, and there has been some recent significant changes in our section staff. And as we are now aware, I am now covering the Northeast region. So we have the Product Safety Network. We provide technical expertise in the safety of fresh fruits and vegetables. We also provide technical assistance for outbreak investigations and compliance and enforcement actions. With this technical assistance, we have the ability to sort of explain the underlying science. A lot of these efforts involve extensive collaboration, both internally and externally. We work with state partners to support product safety rule implementation activities. We also have the Technical Assistance Network that we all know as the TAN. Here, stakeholders and state partners can ask the subject matter expert formal questions about the product safety rule and get official FDA responses. As we know, we also have the regulator TAN support, which is a resource that is available to state inspectors and FDA investigators on demand while they're in the field conducting inspections. So what do we do with all this data? As we receive all these questions through the TAN, the regulator TAN, and the Product Safety Network inspectional data and the readiness review, we collect all this data and we provide an individual response. But also we try to take this information and do other things with it, such as fact sheets, workshops, and guidance. So what happens with all this data? We have been able to identify some common knowledge gaps, of course, through feedback from states and stakeholders, as well as by an analysis of inspectional data. These common knowledge gaps involve subparts E, F, L, and A. And how are we tackling these common knowledge gaps? So we are tackling them by developing additional training programs and outreach materials, as well as revising and updating guidance. For example, for subpart E, F, and L, we are developing some workshops. For subpart E, we are working on fact sheets on qualified exemptions. So workshops. So our division of product safety have been working with stakeholders in industry, academia, and state regulatory programs to offer product safety workshops to growers and other interest members of the agricultural commodity. These workshops are intended to be flexible. Our main goal is to have this workshop offered within the different regions. So as we know, each region has specific conditions and specific practices and challenges, and the idea is to tailor it to where we hold it and with each region needs. We also have now the ability to do this workshop in Spanish for Spanish-speaking audiences, which I can assist, if anyone is interested in doing it here in the Northeast. Currently, these are the 4 workshops we're working in. The Biological Soil Amendment of Animal Origin workshop was created with the purpose and objectives to learn about the current regulatory framework and requirements associated with the use of BSAAO for cover produce on cover farms. We learn about different composting practices within the context of the PSR and about the underlying microbiology of composting and biosolids. During these meetings, we take time to do small group discussions with specific BSAAO scenarios. We know each region has different practices and conditions, so we will be incorporating scenarios that fit within the region. Like, for example, recently we had a conversation about anaerobic biodigesters, so we can discuss how this applies to SOPRF. Another thing that we want audience to learn more is about inspectional approaches and the current status of the BSAAO risk assessment. It's offered virtually, like we did back in December in Florida. We had a virtual composting activity, which included a virtual farm visit, which this was very nice. This workshop is targeted to growers, composters, and other BSAAO industry members, and cooperative extensions and regulators. Now, for these workshops to be successful, we work in collaboration with state partners, industry organizations, like the Produce Marketing Association, United Fresh, National Farmers Union, and the regional and commodity trade associations, and the cooperative extensions. It was most recently virtually offered in Florida in collaboration with state partners, industry organizations, and the University of Florida. And this workshop was meant to be in person, but of course, with the pandemic and travel restrictions, we were able to offer it virtually. Now, here's an example of the flyer that we used back in December virtual workshop event. This is a template that we will keep using moving forward, just to keep it simple and organized. In it, as we can see at the top, it highlights all of the collaborators. Then we have a description of the workshop, the target audience, who is presenting, funding statement, and the topics covered. Another workshop that just recently came out is the Cleaning and Sanitation workshop. The purpose is to make sure that people understand the applicable produce safety rules requirements, food safety risk associated with farm equipment, building and tools, understand the difference between cleaning and sanitizing and its efficacy, the creation of SOPs to manage identified risks, and the importance of activity verifications. Again, we want to tailor it to specific conditions and practices in our region. A good example that is applicable to our region is the practice of using the barrel washer for root crops. We know there's been some talk as of how to adequately clean and sanitize this. And we know that some are well constructed, but there are others that are not well designed to easily taken apart to clean. So this workshop is targeted to farm personnel, specifically those with responsibility for cleaning and sanitizing. So there is a pilot planned in January 2021. It is set to be offered in 6 individual sections once a week for 6 weeks. So this pilot already started January 26th. We already had the first section. The collaborators for the pilot are industry organizations and the Western Regional Center to Enhance Food Safety. This can also be offered virtually, but of course, the goal is to eventually have the in-person option. And here's the flyer that came out for the January pilot section. Again, we like to highlight our workshop partners and keep it simple. The Our Water workshop is currently being developed and it's still in progress. The purpose and objectives are to promote greater understanding of agricultural water. The content will cover water samplings and results, assessing water systems for contamination sources, and we also want to identify water distribution and connection equipment, water treatment equipment options, and assessment resources for determination of water quality. We know the use of water and farm practices may be different from the south than to the northeast, or even within the northeast, there are different practices within states. We also know that every region has their own equipment that meets their needs. So again, we want to tailor this to specific conditions and practices in where it is being offered. This workshop is targeted to growers, regulators, and other state officials. The pilot Our Water workshop was developed, or it's being developed in collaboration with the North Carolina Department of Agriculture and Consumer Services, Industry, and the North Carolina State Extension. Again, this is still in development. And this workshop is not designed to be delivered visually, only because we want it to be in person so that we can see the different equipment. And for this reason, this has been delayed. All right, the Systems Thinking Workshop. So this workshop has been developed in collaboration with FDA, CIPSAN, and ORA Produce Safety Network, and with the Office of Partnership. This is a three-phase workshop, and it is intended for federal and state regulators that conduct on-farm produce safety inspections. Phase one, identify relevant PSR provisions, understand the underlying science, and systematically recognize the public health risk associated with on-farm findings. Phase two, it's going to cover to write the observations and to discuss the on-farm finding with growers. So this will focus in best practices and documentation. The phase three is about looking for compliance and enforcement and implementation. So here in this phase, the participants look at a full picture, and it brings them to start thinking in inspection, compliance, and the outcome for given scenarios. At the moment, we have had 2 pilots in spring 2020, and some more dates are being planned. We hope to have it roll out to states in 2021, but this is yet to be determined. Right, so what else are we doing? The proposed traceability rule is a key component of the FDA's new era of smarter food safety blueprint. It applies to persons who manufacture, process, pack, or hold foods on the food traceability list. Generally, farms will be subject to the requirement in the proposed rule if they manufacture, process, pack, or hold foods on the food traceability list. This also includes entities throughout the supply chain and domestic and foreign entities. We have proposed an exemption for certain small farms, and other proposed exemptions might apply to some farms. So there is a public comment period that has been extended to February 22nd, 2021. And for more information, there are some links provided that we can go. There has been an additional request for information about rarely consumed raw products. And there has been a lot of feedback during the past 5 years and questions about the rarely consumed product list. The issue is that several commodities satisfy the first 2 criteria that are listed here, but the data did not demonstrate consumption of the commodity in any form by at least 1%, which is our third criteria here. So this is a work that's still in development. Other relevant announcements. So the Product Safety Branch has also been working on COVID-19 guidance and other compliance and implementation guidance. For example, on May 2020, an update was announced that the FDA will provide flexibility regarding eligibility criteria for the qualified exemption under the product safety rule during the COVID-19 public health emergency. This guidance is intended to allow affected farmers to shift their sales away from qualified end users while still being considered eligible for the qualified exemption. So this temporary policy is only intended to remain in effect only for the duration of the public health emergency, after which the FDA intends to issue additional guidance. On July 30, 2020, FDA announces a new protocol that was developed in collaboration with the EPA, the Environmental Protection Agency, and it is intended to help companies develop data on the effectiveness of their products in inactivating pathogens such as E. coli and Salmonella in pre-harvest agricultural water. And there's also a checklist that was provided by FDA and OSHA for FDA-regulated human and animal food operations to use when they're assessing operations during the COVID-19 pandemic. So where we're at Subpart E. So the current status, and as we all know, the FDA is working on our rulemaking to propose an approach that offers flexibility and addresses challenges of implementing some of the agricultural water requirements across the diversity in farm types, water sources, and water uses. So this proposed rulemaking will revise certain requirements for agricultural water in the standard for growing, harvesting, packing, and holding of produce for human consumption for covered produce other than sprouts. Throughout this process, we intend to work with stakeholders to find the best way to strike balance between protecting public health and implementing rules that are workable across the diverse industry. We will follow up with more information as it gets released. So in the meantime, FDA encourages farms to keep using good agricultural practices to maintain and protect the quality of their water sources. Those farms that are currently testing their water may choose to continue with their current water testing programs, and farms that are not currently testing their water may choose to start doing so. It is important to note that while this rule extends the compliance dates for the agricultural water provision, that the product remains subject to the other provisions of the Product Safety Rule and the adulteration provisions of the Food, Drug, Cosmetic Act. So that's all. Thank you all again for taking some of your time to hear about our center updates. Please feel free to email me and reach out to me with any questions you may have, or if interested to know more about our workshops, and of course, how we can work together to bring them to your states. With that, that's all I have. Thank you all and have a great day.
Thank you so much, Adriana. And like I said, we will have questions and answers at the end, so please put all of your questions into the chat box. I am now excited to introduce Pat Milner. Pat is a research microbiologist with the USDA Ag Research Service in Beltsville, Maryland. She conducts research in the Environmental Microbial and Food Safety Laboratory and the Sustainable Agricultural Systems Laboratory. Welcome, Pat, and now I'll turn it over to your presentation.
Hi, I'm happy to be here today to talk to you about the work that we've been doing at our Beltsville Agriculture Research Center on the food safety of fresh produce from soil-based production systems. And we'll go through several different things. Some things have to do with soil amendments, and the other part has to do with washing of fresh produce and what we found out about some different alternatives there. The first thing I will talk about is the use of soil amendments, or organic soil amendments, and their different analyses and how they affect the growth of E. coli in a high tunnel study that we conducted at Beltsville. So our purpose was to evaluate the growth and survival as well as transfer of a generic E. coli, it's a marked strains cocktail, to spinach and radish that are grown in certified organic plots amended with heat-treated soil amendments that are basically poultry litter materials. So this was a randomized complete block study conducted in this high tunnel with this sort of plot layout that you see illustrated here. We used poultry litter compost and heat-treated poultry litter pellets, and we compared that with urea. And this was using the kind of methods that we've used for a number of studies throughout the Northeast region, not in high tunnels, but out in open fields. So we were interested in what the survival would be in this kind of a situation. So what we see is from the top graph, we show from day 0 when we inoculated, we had the amendments incorporated, and what did the populations do regardless of regarding the different types of amendments. The poultry litter pellets is the green, the urea is the dark blue, and the poultry litter compost is the red. So we see that pretty typically, as we have seen with open field studies, not in high tunnels, that we can get some increases with these poultry litter pellets, but with the compost materials or this conventional sort of control, the urea, what we see is a gradual decline in the E. coli populations over time until about 40 to 60 days, and then there's a bigger drop-off. Sometimes there's some little tiny blips of increase, but generally it's a downward trend, unlike the poultry litter pellets. Correspondingly, we took soil respiration studies, and what we see is that with these poultry litter pellets, we get a huge increase in soil respiration right away, not unlike the increase that we see with urea, but it's much more pronounced, indicating a greater activity of the overall soil microbiology and the respiration. So when we looked at the increase of what happened over time in terms of E. coli, we see that the heat-treated poultry litter pellets, they supported a really rapid growth increase in a very short time after the material was incorporated into the soil in comparison to the compost and the urea. And this was extended over a very long period of time with the duration. Now, in the National Organic Standard principles and guidance for use of materials, there's an interest in having the compost, or if it's another kind of material, it could be untreated, not heat-treated type of animal manure product incorporated into the soil, but there would have to be a longer wait time. So it depends on whether the commodity is being harvested in 90 days or in 120 days after the application of the product to the soil. If the material is a heat-treated process, like composting, or in this case, the poultry litter pellets, it is a heat-treated process, but apparently the heat treatment has a different effect once it's incorporated into the soil. And so we're really interested in what is it about this sustained increase in the E. coli population in the amended soils when it's the poultry litter pellets. So to look at this a little bit further, we looked at the water-soluble carbon and nitrogen in these amended soils. And what we see is that the amount of soluble carbon released from these soils that were amended with the poultry litter pellets is quite a bit larger than with either the urea or the compost. And the amount of nitrogen is not terribly significantly different from what it is in either of the two more common type of amendments. What we do see, though, is that the amount of soluble carbon per gram of soil in the poultry litter pellets is way higher, but with a vermicompost, it's about in the same range as with dairy manure and with poultry litter compost. So it's the poultry litter pellets, even though they're heat treated, they still generate a huge amount of available carbon. And we believe that this is responsible for the increase in the population. So we went out and we purchased a number of different commercially available soil amendment products, concentrating on poultry manure containing products, and some were advertised as compost, some are only advertised as manures, and then other different kinds that are available. And what we see is that these poultry litter pellets, the heat-treated poultry litter pellets, are among the greatest amount of total organic carbon available in that, this is the soluble carbon that's available in that material of any of these other products. So not all products are created equal, of course. They have also different amounts of nitrogen in them, but it seems that there's something going on with this heat treatment of these poultry litter products that makes them have more available total carbon over a long term. And so what we see is, well, what is the relationship of this total increase in carbon to how, and the extended period of E. coli population, does that actually affect what gets onto the commodity when it's harvested? So we looked at over different days of harvesting, post-inoculation, which is post-amendment as well. And we see that for spinach, we get mean concentrations of E. coli, MPN per gram, or this is based on 30-gram sample enrichment of the commodities. We still get some that are positive out of this. We get less with these composts, a little bit more with the pellets, and quite a bit less, of course, with the urea. But with a commodity that is actually in great contact with the soil, that's where we find that we have the poultry litter pellet is a very high amount here with the surviving, even at the 56 days, which is close to the 60-day time, which we usually see is almost undetectable. By 90 days, all of these are, including the poultry litter pellets, all were negative on the radish bulbs. So this gives us an idea of what we can expect from things that are grown above ground, but in pretty close proximity to these amendments in the soil, and the radishes, which are in really great contact. And the material actually sticks around in the crown area of the radishes. The other project that we have undertaken is to look at what kind of contamination is on tomatoes. And what we saw from a commercial operation on the eastern shore of Maryland is that when the tomatoes are picked, it doesn't really matter which size of tomatoes. Some are the standard rounds and some are the cherry tomatoes or the more oblong tomatoes. The debris from the leaves and the stem get mixed in with the tomatoes that are harvested. And we believe that from the data that I'll show you is that the debris is actually causing some contamination that withstands the chlorination that is used to control the water quality and the content of bacteria in the water. So we were concerned about the problem with salmonella outbreaks that have been recurring over the past number of years on tomatoes. And what we observed in these dump tanks was that tomato fruits are commingled with the harvest debris, which has leaves and stems, as well as soil. And so there's ample opportunity for contact between these materials and also, of course, with the water. So water quality is an issue. Typically, these processors are using hypochlorous acid in the water, and they observe for pH control to keep the active agent at an appropriate level. But because there's an increasing amount of organic matter as all this material is put into the water, it's dumped in, the organic matter increases, soil increases, debris increases, and the microbial load in the water increases. So we're trying to deal with measuring of the free chlorine that's remaining when the conditions are dynamically changing. So we're looking at free chlorine, the ORP, pH, as well as temperature in these conditions. These graphs show that even after 150 parts per million of free chlorine dosing in the water and a 300 micron mesh filtration of the water sample, the tomato fruit microbial counts for these three groups of microbes, Enterobacteriaceae, APC, and yeast and mold, were only reduced about 1 log. And that's up to when it's 150 parts per million free chlorine dosing. In the next series of experiments that were used, they were trying to simulate a tomato dump tank water in order to examine the separate effects of the debris and the tomato fruits as the sources of cross-contamination. So some tomato fruits were indelibly marked to be inoculated with a cocktail of kanamycin-resistant salmonella, and those were commingled in simulated wash water with uninoculated fruits. Then determine the cross-contamination on these different fruits and survival in the wash water. In a separate inoculation challenge, some tomato fruit debris, the green, was inoculated with a cocktail of RIF-resistant salmonella isolates and commingled with uninoculated tomato fruits to assess debris as a potential source of cross-contamination. Salmonella in the filtered and unfiltered wash water with the debris was also analyzed. So there were different ways of preparing the different media, and I can go into that for those who are interested. But for now, let's just get the basic outline here for the experiment. So in this, there's the summary of everything that we found from this showed that the chlorine washing significantly reduced APC and salmonella on the tomato fruits. The chlorine wash was not significantly effective in reducing APC and salmonella from the field debris material, and that cross-contamination of salmonella on the tomato fruits was sporadically observed at very low frequencies at all free chlorine levels that were tested. So in summary, the field debris is thought to be a more important target for reducing the risk of foodborne pathogens in these commingled washing situations. Surface contact between the tomatoes that are contaminated, or tomato or debris that's contaminated, might be a more plausible route of the cross-contamination than that which would be mediated by the wash water itself. So the next step was to look at, well, what was in the wash water where we see that there are these occasional contaminants that do survive in the wash water. And what we see is that this idea of filtering the water through the mesh that's in a Whirlpack bag, or there's a mesh filter there, that filter had, in our case, 300 micron mesh. So it can take that out. But we also tried additional 3 different mesh sizes. And here it illustrates a picture of the size distribution of the particles in wash water. And this is a photomicrograph that shows you the variation in the particulates that are suspended in this tomato wash water. And you see that there's quite a large range, but the biggest effect is with going down to a very small size. When we did a series of microbiome studies looking at, well, if there's no free chlorine, or if the free chlorine is available and adjusted, and if the free chlorine is in the water, but we passage the water sample from the wash water through a 300 micron mesh, or for a much smaller mesh, 63 micron mesh. So we see that if it's a 63 micron mesh, we really reduce the presence of gram-negative bacteria. And that's what you see in these lines that are drawn on the colored portions of the microbiome maps, so to say, and what they're attributable to. And you see that in the one where there's the mesh filtration that's 63 microns, we remove a fair portion of the different groups, and what you have are a tendency to enrich in the gram-positive bacteria, which are characterized by lots of spore formers. So from the tomato wash water particulate studies, we see that basically we have no salmonella that are originating from the inoculated tomatoes where they survive in the wash water, and that some salmonella originating from the debris survived in the wash water, and the bioburden was greater on the larger size particulates. That 93% of the surviving salmonella in the wash water were associated with particulates larger than 63 microns. So cross-contamination from debris to actual tomato fruits was much more frequent than transfer from tomatoes to tomatoes. As many of you may have heard in other types of settings, there are a lot of challenges with conventional immersion tank and flume produce washing systems. This is for cut produce. And this stems from the reuse and recirculation of the wash water. This reuse of water leads to a rapid accumulation of organic compounds in the water, which is accentuated by release of exudate from the fresh cut produce. Several of the organic constituents that are released into the water are major contributors to the rapid depletion of the chlorine sanitizer. So for high throughput commercial production conditions, the dynamic and rapid changes can lead to pathogen survival and cross-contamination, which is clearly something that we want to avoid. A few years ago, a company in South Carolina, McIntyre Produce, offered us an opportunity to conduct some studies at their newly developed and implemented single-pass water produce washing system. And we took advantage of this. We mobilized a mobile lab and set up several areas within their facility where they graciously gave us some sort of sampling and processing area. And we had some sample marathons. And we ended up looking at the microbial analyses from samples and the from this non-immersive fresh cut produce line, and we compared that with the flume line. So what we see here in this picture illustrates the two different kinds of systems. The upper diagram is an example of the single pass line. So the main thing to note about this is that the product comes in, goes through this cutter on the left, and it drops directly down onto a conveyance system. And as it goes upward on that angle of the conveyance system, it's washed. It's washed as it's dropped down, and as it lands on the conveyance system, there's a sprayer that sprays water up, and then it goes through several sets of spray bars, and then it goes through a vibrating conveyance method before it gets to the final wash. The term for this process is called Simply Clean, and the jar on the right shows you the illustration of the Simply Clean water quality that is being used throughout, as opposed to the process in a standard cutter flume system, which is illustrated in the illustration below, and you see the quality of the water that was obtained from that system. And this is in the same facility. So we see that this clean water, Simply Clean system is actually using one pass water. And what they did was they collect the water that's used, and then it goes to a adjacent facility outside of the processing area for water treatment and clarification, a whole multi-step system that allows it to produce essentially potable water. And so these show you some of the differences in the two different systems. On the left is the flume system, which is using the recirculated water over and over through entire shifts, which are 8-hour shifts. And the Simply Clean water, which uses on this vibrating conveyance a spray water that has been through a single pass type system. And this gives very consistent results from each run. We did multiple runs here. So what we've learned about this system in looking at the mesophilic aerobic bacteria, APC, on the final products is that the populations were greatest in the samples collected at the cutter. There was basically no significant difference in the populations on lettuce or cabbage processed using either single pass or the flume system. So they were pretty close in agreement, but you have the consistency with the single pass system that the water starts out each time the new batch of produce goes through the cutter, it's getting fresh, clean water, not recirculated water. So the key findings from the comparisons of these flume and single pass system is that they offer more consistent water quality with the single pass system because you're not using the recirculated water. There's a reduction in the accumulation of organic compounds in the water, and so it's easier to control the processed water conditions. They do generate comparable APCs on the final products, and they give slightly better sensorial results after storage over a 3-week period with the single pass products. The critical step seems to be the cutter, and that the organic removal and the microbial safety control potential at that cutter from using clean water instead of recirculated water is significant. And we hope to be able to do some additional future studies on this types of systems. So we had quite a team of people doing all of these different types of work, and we are very graciously appreciative of the people at McIntyre Produce for allowing us to perform these studies. And the reference that's available here, you can get additional information about the details of these studies. I've only been able to go over a very rudimentary way. There's one more thing that we are embarking on, and I'm going to show that in the last slide. So the next venture that we are undertaking is on the system called aquaponic production, which uses fish aquaculture in container tanks. And we have our aquaponics system, which is water recirculating to a grow bed, which is up on the top part of these pictured units that you see here. We have our 8 replicate 130-gallon recirculating aquaponics system research units located in a high tunnel on our north farm at Beltsville. In addition, we have 3 replicated decoupled aquaponic research units also in a high tunnel at this facility. The decoupled system allows for basically a separated treatment system to treat the water from the fish tank before it enters into the plant production portion of the system. It allows for some adjustment of micronutrients that might be needed to supplement for plant nutrition. So we're venturing into this with a number of different partners, and this is our next avenue because there are really no good metrics for survival of pathogens and other parts of the important microbiome that help with the treatment of the fish wastewater. So with that, I'll close and thank you for your attention and invite your questions now or at a future time should you want to explore anything further. Thank you.
Thank you so much, Pat. I really appreciate it. And now we'll take some questions and answers. So we'll start. There's one in the chat box for Adriana. Adriana, is there a process to workshop the workshops that you mentioned with farmers of various scales to ensure that they're helpful and meeting the desired objectives?
Hi, and to answer your question, so yes, so our workshops are designed to be flexible to any scale, small, medium farmers. Our goal is to have these workshops offered within the different regions, as I mentioned in the presentation. And we know each region has these specific conditions, and each grower has specific practices, challenges, and of course, different objectives. So we try to accommodate to each region needs. Yeah, so this means, you know, like we know that the product safety rule is also written in a flexible manner. So this means that the rule has to also be applied and interpreted in light of region-specific conditions and practices.
Thanks, Adriana. The next question is also for you. It comes from Amanda Kinchlet, University of Massachusetts. She noted that she's remembering a team in the Western Center developed an ag water training program funded through a USDA FSLP project, and was wondering how the PSN workshop is similar or different from that.
Okay, first, good to hear there are discerning opportunities out. We all have the same goals, and this is to educate and work together with stakeholders in industry, academia, and state. Of course, in providing education and to promote, in this case, greater understanding of ag water. How does it differ? And here we have to go back to the differences between regions. Like, for example, the equipment each region uses. We know that every region has their own equipment and meets their needs. So, with our workshop, we want to tailor this to the specific conditions in practices. In this case, to the practices and conditions here in the Northeast.
Thank you so much, Adriana. The next question is for Pat. This comes from New Hampshire. Pat, in a recent high tunnel fertility study Heather participated in, they found that because you are basically creating an artificially arid environment in a high tunnel, some fertilizers did not dissolve or break down the way that we were used to them dissolving or breaking down in field soils. Has the high tunnel study been replicated outdoors, or is there any intent to replicate it outdoors?
No, we have not replicated this outdoors, and at this point, we aren't intending to do that. We really did not experience a lot of a problem with letting the plots dry out because we were pretty attentive to watering them very, very frequently, and the moisture levels were always maintained at a pretty high level. So they were not. There was not any drying out or wilting of the plants, and the soils were kept pretty moist all the time. These were artificially, watered. We had water that we got from our system that's on that North Farm, and we put it in a reservoir to ensure that it was dechlorinated before we irrigated it with sprinklers.
Thanks, Pat. Another question for you coming out of Maine. The question is, sorry, I lost it here. So just out of curiosity, how were the inoculated versus uninoculated tomatoes marked that tell them apart as you measured cross-contamination in the uninoculated?
Sure. We used a lab marking pen. These are the indelible Smarty markers with a black mark across the laterally on each side all the way around the ones that were inoculated. So those, when they dried, they didn't wash off.
Thanks so much.
The next question for Pat is, should water be single use then?
Well, based on our studies with these large volumes of tomatoes that we saw in the commercial sector, I would say it really depends a lot on the volume of the water flume, how much they're using in that wash water, and whether you're having a lot of debris that goes in with it. I think that the situation with a lot of the commercial growers and commercial operations, when they have people picking, especially with a lot of the smaller tomatoes, there's a tendency for the people who are doing the harvesting to pull the whole group and cluster of tomatoes, which in turn brings into it a lot more debris than if you're picking individual fruits. So whereas in your backyard, you might go out and pick individual fruits, but they're working to do a job in a hot weather usually and quickly. So there's a lot of debris that gets in there. So the other thing is the total amount of tomatoes that you're using in that volume of water. So we didn't try to scale this down for smaller batches. We were just doing a simulation, and that's the very smallest that we could do, which was a lab scale situation, just to find out the purpose of whether it's really being from tomato to tomato, or is it the debris that's really there, or is it left over from what's in the water? And it turns out that the particulates that get released, and a good amount of those particulates come from the debris.
Pat, the next question is also for you. It is about whether the level of APC measured was determined to be within an acceptable range for the products tested.
Yeah, we didn't really try to target or extrapolate to what might be an acceptable range. The feature that we were looking at is once we had subjected the tomatoes or the debris and the combination of those to the chlorinated water, once we understood about these particulates in the water, it's really more about what is the type of organisms that we're seeing. And what we saw was that we're getting a proliferation or, resistance by the gram-positives and not so much the gram-negatives remaining. So there seems to be a differential. So if you just go by APC counts and you don't try to separate that out, then you miss part of the story there. So I think more has to be done to really follow that up if you want to get to an acceptable count range.
Thanks, Pat. One more for you. Will the aquaponics study look at evaluating the appropriate indicator organisms for water quality testing and food safety?
You betcha. That's a big issue for us, and one that we want to tackle, and not just for water quality, but for what may actually get transferred to plants one way or another. And this involves a lot of practices, involves issues of the aquaponics design and the practices that are actually used. And there's different ways that the aquaponic plant side can be configured. So we have to look at a variety of those different things. So yes, and there's a variety of different organisms that one may not normally think about.
Thanks, Pat. Adriana, back to you on some of the workshops that the PSN is coordinating. The question is, how will the PSN workshops be delivered within state jurisdictions? And there's the second part of that is, will this be coordinated with state programs under Comp A activities? So I think looking for some state coordination.
Yeah, so we know, we understand, we know each state has different jurisdiction, and of course we will work around that. Again, these are designed to be in coordination with different state programs and stakeholders. So we will work with the different states individually and find the best way to bring them over.
Thanks. And I just want to add Stephanie Brown from the Western European Center, who is joining us at a very early hour, which we appreciate. Also noted that they were involved in the pilot workshop on cleaning and sanitizing, and that the audience was specifically limited to folks in industry involved in cleaning and sanitation activities, just with an intent of getting that industry stakeholder feedback as part of the pilot process. Next question is Robert's, Elizabeth.
Yes, thank you. So Pat, back to you, a question about the tomato study. Are there any plans to, in the future, look at sanitizers other than chlorine? I'm wondering what the results might have been if something like PAA was used rather than chlorine.
Right. We don't have any plans right at the present time. We're certainly aware of the use of PAA and the interest in using that, but we have not put that on our schedule of the next things to do. So it's something that we could do, but it isn't one of the primary things right at the moment.
And also related to your presentation, Pat, is with respect to pelletized chicken manure, is the thought that the heat-treated pelletized chicken manure will break down more quickly in the soil compared to the other soil amendments assessed, providing a more conducive environment for the growth and survival of pathogens?
It's more that not necessarily the heat-treated pelletized. It's probably if every other product that we looked at was pelletized or pulverized, which is what happens to the pelletized material before it gets pelleted, that would make sites more available for microbial breakdown and release of other kind of nutrients and possibly other processes, transformation processes to occur. Our concern was that the heat treatment itself, after the material was pulverized, is that there's something going on there. And if you've seen these pellets, you'll see that once the water hits them, they just fall apart. However, sometimes they don't fall apart so quickly, and we were wondering if people are using some kind of binder in them, and if there's a binder added, if that binder has some particular soluble carbohydrate that might act as a suitable microbial nutrient very rapidly, that could be part of it. The other part of the whole heat-treated pelletized material is that there doesn't seem to be a lot of control and calibration of these heat treating processes. I think there are a lot of different vendors around that are doing this, and some have a little more conscientiousness and calibration control over their process than others. And I think that process control, just like composting, is going to make a big difference in the quality of the material relative to what we're talking about, and whether that material can inherently support a microbial burst in population, if you will, once the material is wet and releases into the soil those nutrients.
Thanks, Pat. And we have one more question, and then we'll close out this session for this morning. But this is again for you, Pat. From Sean in New Hampshire, most of the experimental aquaponics systems that he's seen are still based on the UVI system design from the 80s. In particular, folks don't seem to be worried about retaining lots of organic carbon in their systems, which to him is problematic. So at UNH, they tried very hard to minimize total organic carbon accumulation. Can you tell us what kinds of solid removal you're using in your system?
It's a biofilter system, so there's little plastic, there are little plastic knobs. I don't know what else to call them right now, but and there's aeration in that, so it's an aerated biofilter activated system. So it's and there's filtration with that too. So it's kind of multiple steps. With the idea is to remove a lot of the solids, but to allow it to break down to its components first, but then to convert that into nutrients, have a digestion process and transformation, nutrient transformation.
Great. Thank you so much to both of our presenters this morning and for taking the time to answer these questions. I really appreciate it. And I appreciate all of the great conversation we had in the chat box. I'm sure that if you have additional questions that you can email the presenters and get information from them directly. So thank you again. And now we will go into our concurrent work group meetings, produce safety and preventive controls again this morning. And I appreciate all of you being here and look forward to continuing our discussion. Thank you so much.